Breaking the wall of small animal imaging. Stanislav Hrivnak, Pavol Josef Šafarik University in Košice, Slovakia.
So probably most of you have heard about CT devices, right? These days they are commonly used in hospitals, and you can perform a 3D imaging of your chest or your brain, and maybe some of you even experience this. It looks like this. But the thing is that sometimes you want to image something that is pretty small, maybe in a millimeter range or even smaller, maybe some cell or some microorganism. And these devices exist. They are called micro-CT globally. And it turns out that it's much more challenging to perform micro-CT than regular CT. And here in the image, you can see how does it work. So you have basically some X-ray source, you have some small sample, it hits the sample, then it goes further to some optical element that's very important, and then it reaches the camera or the detector. And what we were asking is there some better way how to perform micro-CT? And we believe that yes, there is. And our solution, we called it tuned Bragg magnifier, so you can see in the image. And the idea is in the optical elements. So these four black parts, these are optical element, and it's crystals. These are germanium crystals. And the whole point of them is that they are magnifying the beam, magnifying the X-rays. And because they are magnifying it, you can use detectors which are bigger. And bigger detectors are much more efficient than smaller detectors. So the whole key point of this is that you can image super small things without the need of super small detectors. And you can be much more dose efficient, so you don't want to kill your sample with X-rays, for example. So my contribution in the whole of this project was to build a mathematical model of the optical element and to build a 3D reconstruction algorithm. So here you can see, for example, the comparison between the old ways how to do it and my solution right now. And we applied this to the 3D imaging of a small organism called tardigrade or water bear. So for example, here you can see it. The size of it is approximately 100 micrometers, and we performed this in a resolution of around 300 nanometers. So this was all done by this tuned Bragg magnifier with our new algorithms. So thank you very much.